So in this experiment, we were supposed to get the DNA out of a banana. And well, at least in this part of the experiment. And so here it is. We needed to use some alcohol, dish soap, and salt to separate the organic material from the DNA strand. And here you can see that the some of the materials that you're going to use are, as I said, dish soap, banana, ethyl alcohol, water, um, some coffee filters, funnel. Also, you're going to need somewhere to put the banana and mix the ingredients together, and um, something to smush the banana into somewhat of a jam or paste, and somewhere to collect the organic material once it starts the process of separating. So what you're going to do is first take your banana, put it in the place that you're going to smush it, then grab whatever you are going to use to mix the banana with the other ingredients and smush the banana, smush it as good as you can, then put the dish soap, then put half a spoon of salt, and then put some water. So you're going to start mixing it till it's somewhat of a liquid. After that, you're going to put the coffee filter inside the funnel, and on top of the coffee filter, you're going to put the mixture that you just made, the banana mixture, and put some water in it so it starts dripping from the coffee filter. Use something to move the mixture around so that it filters faster, and then you collect the whatever it's going to pass through the coffee filter down the funnel. And after that, you're going to see the results of the experiment, which are here. You're going to see the organic material. The dish soap is not visible because this is yellow, but the kind of cloudy material is the DNA. Now, this is important because with this process, we can already understand how we can get information from uh, cells, and with that DNA, we can now analyze it so we can understand the genes that make up the organism. This is a really important process for any genetic engineering that you might want to do with the manipulation of genes.
On the second experiment, the main DNA that we were going to use was the saliva. That is the main ingredient for this, and we were going to use it for analyze what's inside of it and what was its effect. And the main ingredients that we used for this was the salt, the saliva, the dish soap, and the ethyl alcohol. All that we need to do is what it was putting all the ingredients in small amounts and then mix it with a swab. And when the mixture was done, we have to see what was its effect, and we see that the main effect was that it was transparent, and that is because of the saliva and also for the ethyl alcohol. In conclusion, we can say that getting out the extraction of DNA in some living organisms, that in this case were fruits with homemade materials or even the laboratory material, can really help out to analyze what its content, and it also helps with the molecular biology. And we think that this experiment really helped us out to analyze what's really inside the living organisms like the saliva and the fruits.